Thanks for the invitation for this great symposium. My lab works on Streptococcus pneumoniae, which is quite an interesting gram-positive opportunistic pathogen. So the natural niche is the human nasopharynx. So that's the cavity right behind your nose. There it colonizes people, mainly young children up to the age of 5. This is a very interesting environment, polymicrobial environment. A lot of different bacteria are there, and they all compete for host-derived sugars. And when pneumococcus, is present, is colonizing you, it's basically not a problem. But once it moves to a different niche, like the lung or the blood or the brain, it can cause really serious problems and invasive diseases such as pneumonia or sepsis or meningitis, killing more than 1 million people every year. So this is a really serious problem. So another serious problem is that we see an increase in antibiotic resistance. So multi-drug resistance is on the rise. So it's clear that we need new antibiotics. But what is very interesting, especially related to pneumococcus, is that certain antibiotics activate competence development. So we've already heard from Melanie what competence is, but just to summarize very briefly and why this is interesting for pneumococcus. So cells can become competent. Then they can take upon a certain, they can become competent upon a certain environmental signal, and then they can take up free DNA, and then they can integrate this and they can become transformed. So now imagine that in the case of pneumococcus, certain antibiotics act as the environmental signal, and now imagine that this free DNA can encode antibiotic resistant marker, then you end up actually with an antibiotic resistant bacterium. So this is quite intriguing. And this is something we were interested in studying. So this phenomenon was first discovered by the group of Jean-Pierre Claverie in Toulouse. And they showed in this essay here, using a firefly luciferase reporter under control of a competence-specific promoter, that certain antibiotics like mitomycin C and fluoroquinolones induce competence. And you can see that here by the expression of light. And this is depending on the competence regulatory network. So we had some hypotheses how this could work. We thought there might be a link between the cell cycle and competence. So we wanted to explore that further. But first we need to know a little bit about the pneumococcal cell cycle. So what do we know? We know it has a single circular chromosome with the origin of replication and probably where replication starts bidirectionally from. We know it must divide somehow. But first, to get some idea about the pneumococcal cell cycle, we developed some tools to look into this in more detail. So here's just one of such an experiment where we made a double-labeled strain where we fuse a component of the DNA replication machinery to green fluorescent protein, and at the same time we fuse FTSZ, so the cell division protein, with the red fluorescent protein. And we make these types of movies, which we then analyze to get an idea of the pneumococcal cell cycle. So what you can see here is that these green dots, they jiggle around. So these are the replication machineries, and in red are the new division sites. And I hope you can appreciate that the green dots sort of jiggle around the middle of the cell here, but are quite dynamic. So of course then we gather a lot of these images and we need to quantify this somehow. So for that we developed our own analysis program called Spot Process R. It's not relevant for the talk now how that works, but we can really do quantitative analysis of these microscopy images. And things that we can do are, for instance, make subcategories of groups of cells. So we don't have to take specifically pictures from time-lapse microscopy. We can also grow cells from a balanced growth. And then the cell size is actually a very good proxy for the cell cycle state. So if they are very short, it means they're newly born. If they're a bit longer, it means they are more advanced in their cell cycle. And we can make, for instance, 4 subcategories. And then we can look at the localizations of thousands of cells in these cells and make, for instance, these heat maps. So here's the overview of FTSZ. And what you can see here also in these histograms is that it's very nicely localized in the middle of the cell. So here's the outline of the cell. And you can see in these pre-divisional cells that FTSZ moves to the new future cell division sites. So that's nice and what you would expect. This is now the picture for the replisome, and you can see that it's also roughly localized in the middle, but you see the spread is much more broad than FTS said. So meaning, as you could also see in this movie, that it's quite dynamic. So we can also label specific loci of the chromosome to see where the chromosomes are and how many chromosomes there are. So this is another type of graphical picture of these types of data. So here we labeled the origin of replication in green, and now here on the x-axis is the cell length. So these are short cells, these are long cells, and this is sort of the location from the middle of the cell. So you can see the origin splits very rapidly in the cell cycle, and then the two new daughter cells end up with one origin of replication. And in the same strain here we localize again the replisome, so you can see it's nicely localized in the middle of the cell. So this already shows that we don't have multi-fork replication like you have in fast-growing E. coli or bacillus. We just have a single chromosome with two replicating origins of replications that segregate pretty quick during the pneumococcal cell cycle. So we did a lot of stuff more, and I don't have the time to go into detail, but now we have a good idea about the pneumococcal cell cycle. And basically it starts by initiation of DNA replication, more or less in the middle of the cell. Then the origins split and segregate to the future division sites. Then the left and the right arms are segregated and also move to mid-cell. And then finally the TER stays in the middle and segregates just before cells divide. So now we know a little bit more about the pneumococcal cell cycle. Coming back to the question about competence. So I have to explain a little bit what we know about the gene regulatory networks driving competence in pneumococcus. So at the heart of the system you have a two-component system, COM-D and COM-E, and a small peptide, COM-C, which has a leader sequence which is then cleaved off by an exporter, COM-AB. This is a double glycine motif which is recognized. Then COM-C comes outside the cell, and then we call it CSP for competent stimulating peptide. And then once a threshold level of CSP has been accumulated, this is sensed by the histidine kinase COM-D, which sits in the membrane. This then autophosphorylates, donates its phosphate to COM-E, the response regulator, which then dimerizes and activates a number of genes, including its own operon. So this is a positive feedback. So once this threshold level of CSP has been reached, all cells effectively express competence and become competent. COM-E also activates COM-X, which encodes an alternative sigma factor, and this then switches on the genes required for DNA uptake and recombination. So for the talk, you just need to remember, if you have more of these COM genes, you become competent. So now, since we now know a bit more about the pneumococcal cell cycle, we had this hypothesis about how these antibiotics induce competence. And that was because basically we saw that these COM genes are very closely located to the origin of replication. So we thought perhaps these antibiotics like mitomycin C and fluoroquinolone, they might stall DNA replication. And then we tested that by using a compound called H-pura, which stalls DNA replication elongation. And the idea was then, if you stall DNA replication elongation, but don't stall new rounds of DNA replication initiation, you end up with a situation that you have multi-fork replication. So in this case, this cell would have 4 copies of the origin versus 1 copy of the terminus, whereas this cell would have 2 copies of the origin and 1 copy of the terminus. Now, since these COM genes are very closely located to the origin of replication, this cell will have more COM genes, and because of the positive feedback loop, it will switch on competence. So to test this, we perform genome-wide marker frequency analysis. So here we have the circular chromosome. Now we cut it at the terminus, so the origin is here in the middle. And we plot the copies of genes of cells treated with H-pura versus untreated cells. And what you see is that indeed genes close to the origin of replication are more abundant relative to untreated cells. So you have more copies of these genes close to the origin. And of course, we can also look at the transcriptome. So will you get more transcripts when you have more copies of the DNA? And this follows suit nicely as well. So indeed, this works, and this is a control with an antibiotic that doesn't induce this effect. So to show causality, we played a bit around with the location of these comp genes. So this is the natural situation. So we use the same trick as Jean-Pierre Claverie. So we have a firefly luciferase here, and we give now H. pura. So the comp genes are here, close to the origin. This is our luciferase construct. So we give antibiotics, so the cells grow a little bit slower, and they produce light and actually take up DNA, so they become competent. So if we now move away the comp genes from the origin at about 90 degrees of the circular chromosome, and we add H. pura, competence is no longer induced. If we put a second copy of the comp genes, we see already earlier in growth that these antibiotics induce competence. And when we put the second copy also really close to the origin of replication, now even without antibiotics, cells already become competent. So this shows that really the origin proximal location of these comp genes triggers antibiotic-induced expression. And this is not only true for H. pura, but for basically all antibiotics that affect DNA replication. So that's what I just showed here, and especially in the case of pneumococcus, since these comp genes are really closely located to the origin of replication within 5 kb, they are activated. Okay, so this we published now already 2 years ago, but we did a lot of RNA sequencing for this project, and one thing we noticed is that all the antibiotics that induce competence also induce expression of bacteriocins. And antibiotics that didn't induce competence didn't induce expression of these bacteriocins. So what are these bacteriocins? So these are basically toxins that kill other bacteria. I already told you that pneumococcus lives in a very interesting niche, polymicrobial niche, where they have to compete for these nutrients. So now we have the situation whereby you give antibiotics, and this actually activates these toxins. So how does that work? So what do we know about the gene regulatory networks driving these bacteriocins? So the system looks very similar to the competence system, actually. So also here we have a small peptide called BLPC, which also has a leader with the double glycine motif, very similar to the COMC, which is presumably exported by an exporter called BLPAB. Then it's processed, then BLPC accumulates, and upon a certain threshold level of BLPC, the histidine kinase is autophosphorylated. It donates its phosphate to the response regulator, and then BLPR, the response regulator, activates the bacteriocins. So those are these small genes here, but also the immunity genes. So the cells also become immune to their own bacteriocins. Okay, but we had a problem because the strain we work with, the D39 strain, has 2 frame shifts in this exporter. So that was the first puzzle. So we wondered if our strain is perhaps a bit peculiar or funny. So for that, fortunately, we have a lot of pneumococcal genome sequences around, and we looked at them. And actually what you see here is that most of these pneumococcal strains actually have a disrupted BLPAB. So all the ones here in black are disrupted, and only the ones in green have an intact BLPAB. And actually it's about 23%. So for the rest, you have all kinds of variants where you have whole pieces missing of BLPA or of BLPB or frame shifts, etc. So it doesn't seem that our lab strain is weird. But actually in the literature it has been described that these strains that have a disrupted BLPAB are so-called cheaters, because they don't have to actually secrete the peptide, but they can sense the signals from other bacteria. But if you think about it, that only 23% of all pneumococcal strains can do that, then that's a bit weird if those are the cheaters. So more evidence that these strains still can produce bacteriocins is that if you look at the number of bacteriocins that strains harbor, this is more or less the same in whether strains have intact BLPAB or disrupted BLPAB. So the median is about 5 bacteriocins per genome. So there's no genetic drift here going on. Okay, so how is this regulated then, and how do antibiotics actually activate these bacteriocins? So there are 5 promoters driving these genes, these BLP genes. And what we did, we took all the promoters and cloned them in front of firefly luciferase, and then we looked at their gene expression. And what you can see here is, here in black, that a nice pulse of gene expression of these BLP genes really at the end of exponential growth. And this is really dependent on the two-component system. When we knock out the response regulator, you don't have activation of the Bacteriocin genes anymore. This is true for all for these 4 promoters, except for the promoter driving the two-component system itself. So this is a constitutive promoter. So we also notice is actually that the expression of the Bacteriocins is pH dependent. So, and this is very similar to what we have in this situation of competence. So in more acidic pHs, the Bacteriocins are not expressed, but at pHs above 7.4, the system is switched on. And indeed, if we now take cells at more acidic pHs, we can induce the Bacteriocins by adding antibiotics. So this is in line with what we saw with our RNA sequencing data. But this is very similar to what we observe with competence induction. So to figure out if there's a sort of interlink between competence and Bacteriocin expression, we made a double-labeled strain. So here we fuse a competence-specific promoter driving a red fluorescent protein and a Bacteriocin promoter driving green fluorescent protein. And we spot cells and we do time-lapse microscopy. What you can see after 30 minutes, basically all cells become competent. And then about 150 minutes later, it's hard to see here, cells also express the Bacteriocin. So there's a serious time delay here between switching on competence and switching on the Bacteriocins. Okay, so we wondered perhaps switching on competence can directly activate the Bacteriocins. So we can activate competence by adding synthetic CSP. So this is an experiment where we take a competence-specific promoter, we give CSP, and immediately you see competence. And when we do that and we look at Bacteriocin gene expression, we can see that also CSP can induce Bacteriocin gene expression, but again we have this 100 to 150 minutes of delay. So adding synthetic CSP can activate Bacteriocin gene expression. So, and if we zoom in, you see a little peak here, now we zoom in. This doesn't depend on the Bacteriocin two-component system. So when we knock out the Bacteriocin two-component system, competence is, CSP can still induce the Bacteriocins. However, this strongly depends on the competence two-component system. So in the absence of comp-CDE, you can add CSP, but you don't have this small but significant increase of Bacteriocin gene expression. So this shows that expression of the Bacteriocins depends on both comp-CDE and BLPR-H. So this still brings me back to the question how actually BLPC is exported, since we have these frame shifts in BLPA and B. So we now know that once cells become competent, COM-E can activate, of course, the competence genes, and then and at the same time also activate these BLP promoters. So since it seems to be so similar, we thought perhaps actually COM-AB can take over the export of BLPA-B. So if we look now again at these 4,000 genomes, we see that all strains have an intact COM-AB. So that's the two outer rings in this phylogenetic tree. So whereas 75% of all the strains have disrupted BLPA-B, all strains have an intact COM-AB. So, well, this begs the question whether COM-AB can be the exporter for the BLPC peptide. And indeed, that's the case. So when we knock out COM-A or COM-B, Bacteriocins aren't expressed anymore. Only when you have both COM-A and COM-B, Bacteriocin gene expression is switched on. We can complement this, so if we induce ectopically COM-AB in a COM-AB mutant, again we can get Bacteriocin gene expression. So together this gives us this interesting regulation picture, I think. So antibiotics can induce competence, for instance via this gene dosage effect. So COM-C is built up, CSP is accumulating, this then switches on COM-E, which then activates all the competence genes and the DNA uptake machinery. At the same time, COM-E slightly activates the Bacteriocin gene expression. Importantly, also the expression of BLPC. at low levels. So BLPC is then made and then is exported by COM-AB. And then once enough BLPC has been accumulated, this is sensed by BLPH, which then autophosphorylates BLPR, and then the Bacteriocin genes are highly activated, explaining this temporal delay that we see. Okay, so to conclude the story, basically we've showed that COM-E activates BLPC expression. BLPC can be exported by COM-AB, and I think this data discards this cheater hypothesis, since most strains actually have inoperative BLPA-AB and can secrete probably bacteriocins via the competence system, which all strains have. What's quite interesting is that bacteriocin expression thus requires accumulation of 2 peptide pheromones, so both CSP and BLPC, which is so far quite unique. But the bottom line is, since antibiotics induce competence, they also induce bacteriocin expression. And this is also a bit in line with what we heard from Kevin Foster, that cells are a bit stressed, and in response, actually, they switch on toxins, for instance. So what this means, well, we don't really know. You could argue that perhaps after antibiotic stress, well, making bacteriocins to kill other bacteria might help to clear the niche even further. You could think about the use of the free DNA to acquire, for instance, antibiotic resistance, or food, or basically resist mobile genetic elements. That's also a hypothesis. But in any case, I think it's fair to say that antibiotic treatment for pneumococcal infections can have unpredictable consequences on the whole colonization dynamics, because you thereby also activate these bacteriocins. All right, so this work has been done, so all the microscopy work I showed you was done by Renske van Raaphorst, the competence story done by Jelle Slager, and the bacteriocin work by Morten Kjos, who just started his own group now in Oslo. And with that, I'd like to thank you for your attention.